Assalamu alaikum. Today I'm going to tell you about what is Onco Query Language, which is very important to know. Onco Query Language defines the specific types of alterations to be considered when running a query and why it's necessary when you run a query on one or more genes. Onco Query Language defines which genomic alterations count towards a sample being altered. What does that actually mean? Let's look at an example. Okay, firstly, you have to enter your query. Okay, what happens in a regular query? This query looks for sample with alterations in IDH1, IDH2, EGFR, and T53. These are gene names. We can see that the presence of any of three different alterations, amplification, deep deletions, or mutations, define a sample as having an alteration in a query gene. But how were those three types of alterations selected? How do we know if an alteration isn't present in the data or just isn't being examined in this query? For example, are there samples with fusions or shallow deletions in any of the query genes? If you hover over a gene name, you can see the specific alterations which were included in the query: mutation, fusion, amplification, or homodeletion with inverted commas. These are the default Onco query language options and will highlight any mutation, fusion, amplification, or homozygous deep deletion in the query gene. So let's come back to this question. Are there samples with fusions or single copy deletions in any of the query genes? Because fusion is included in Onco Query Language but is not shown in OncoPrint, we know that there are no reported fusions involving these genes in the data uploaded to CBAR portal for this study. An important caveat here is that many studies do not standardly report fusion, so the absence of data does not necessarily reflect that fusions are not truly present in these samples. Shallow relations were not included in Onco Query Language for this query. So there may be shallow deletions affecting these genes, but we won't see them because the query didn't look for them. What if we want to include shallow deletions? How do we do that? Let's learn how to use Onco Query Language. Rules of Onco Query Language. Onco Query Language uses keywords to define the alterations to include in a query. To write is a table defining the journal, keywords, top, and the modifiers which can be applied to certain keywords in the bottom. The complete specifications can be found here. So let's look here. These are mutations and their keywords and syntax, and these are fusions with keywords and syntax. These are copy number alterations which can be amplification, deep deletions, gain, shallow deletions, and these are mRNA expression with their keywords and protein phosphoprotein level with their keywords. These are keywords, driver, germline, somatic, and applicable data type is written over there. So let's recreate our initial query. On the left is the query as we ran it before. On the right are two different ways to write the exact same query using Onco Query Language. So on the right side, you can choose any cancer sample and then you have to enter your query genes. The journal format for Onco Query Language is gene alteration 1, alteration 2. But as shown in the bottom example, the data types command allows a user to select the same set of alterations for multiple genes all at once. So here each gene is listed on its own line, followed by a colon, and then the list of alterations. Rather than writing the same alterations after each gene, the data types command can be used to set the alterations for multiple genes at once. Now let's adjust default query. Let's look for gains in EGFR and shallow deletions in TP53. Add gain and het loss to the query. OncoPrint now includes gains in EGFR and shallow deletions in TP53. What if we want to look at IDH1, R132C mutations but no other IDH1 alteration? We can specify the mutations in Onco Query Language. We then see that there are many fewer examples with mutations in IDH1 since we have limited the query to relatively rare R132C. We can further refine the query by removing alteration types that are not biologically relevant, like the deep deletion in IDH2. Okay, let's take an example of BRCA1. Loss of BRCA1 is a common event in ovarian cancer. What percentage of samples lose BRCA1? Let's run a query to find out. Looking at OncoPrint, we can see that 12% of cases have an alteration in each of BRCA1 and BRCA2. However, this includes amplifications which will not result in a loss of function. We can use Onco Query Language to make the query more specific. Modify the query to include only mutations and homozygous deletions. OncoPrint now shows a more accurate estimate of frequency of BRCA1-2 loss. However, mutations and deletions are not only way to decrease the levels of functional protein in a cell. DNA methylation can lead to decreased mRNA expression. We can use the Flower step to examine the relationship between DNA methylation and gene expression. Note that the lower right quadrant contains samples with low expression and high DNA methylation. We can also use Onco Query Language to identify these cases. Modify the query to also include samples with decreased expression. Don't forget to select mRNA expression in the Genomics Profile section. OncoPrint now shows a more accurate estimate of frequency of BRCA1 loss. Some mutations in OncoPrint are variants of unknown significance. Recall from single study query tutorials that mutations are annotated as drivers or unknown significance based on mutation drop down. We can further refine the query by only including those mutations which are putative drivers as defined by mutations dropdown. We can do this by adding driver to mutation term in OncoQuery language. This will include only mutations that are putative drivers. Or replace the entire OncoQuery language string with driver. This will include mutations, fusions, and copy number changes that are putative drivers. See BRCA2 below. Compare the result of this latest query in top with the previous query in the bottom and see that the mutations of unknown significance are no longer present. This study is one of few in C Bioportal that include germline mutations. We can make one final adjustment to our query to ask a slightly different question. What percentage of samples have putative driver germline mutations in BRCA1, BRCA2? Note that the OncoQuery language for BRCA1 and BRCA2 are equivalent as germline term only applies.